So our first speaker is Rafia Akhlaq. It's my great pleasure to introduce her. She's currently a master's student in molecular medicine in University of Karachi in Pakistan. She has received her bachelor degree with honor as well as master degree in microbiology in the same university. And she has received multiple awards as well as scholarships, including the prestigious Regeneron Scholarship. She has also done a lot of teaching to kind of promote the diversity as well as gender equality in Pakistan. And she's also a freelancer academia writer at TrueLancer. So just everyone know that she's actually open for PhD opportunities in the USA. So if any PI has any opening, or if you know anyone has PhD position opening, please feel free to reach out to her directly or write us an email so we can also forward to her. With that, I would hand the floor to Rafia to tell us about histone modifications in squamous carcinoma.
Thank you. Can you hear me?
Yeah, we can hear you.
Okay. Thank you for the nice introduction. I am Rafia Akhlaq, going to present my master's research work, histone deacetylase and thioredoxin inhibitors regulating histone acetylation and methylation levels under hypoxia in oral squamous cell carcinoma in vitro. In oral cancer in Pakistan, it is a major problem of developing states, according to Globocare 2020 report. Oral cancer is the second leading cancer in Pakistan. Among many therapeutic resistance in oral cancer, we will focus on hypoxia and epigenetic changes. Hypoxic tumor microenvironment regulates chromatin in various ways, including histone regulation in histone modifications, mainly acetylation and methylation. Histone deacetylase inhibitor that we have used in our study is voinostat. It is a small molecule inhibitor and an FDA-approved drug for cutaneous T-cell lymphoma. It targets hypoacetylation in cancer and inhibits histone deacetylases, causing accumulation of acetylated histones. In results, cell undergo apoptosis. Unfortunately, histone deacetylase inhibitor have shown some resistance in cancer, particularly in solid tumors. It have shown low therapeutic efficacy as a single agent and have shown good therapeutic efficacy when used in combination with other anti-cancer agents. Histone deacetylase inhibition is linked to thioredoxin inhibition. We hypothesized to target this thioredoxin inhibition in combination with histone deacetylase inhibition to target cancer cells. The thioredoxin inhibitor that we have used in our study is PX12, which have gone to clinical trial for pancreatic cancer. PX12 inhibits TRX1 thioredoxin protein and inhibits its activation through thioredoxin reductase enzyme. Coming to research objectives, we have mainly two research objectives: testing individual and combination efficacy of voinostat and PX12. But however, we will focus on this objective that is relevant to histone modifications, that is testing effects of voinostat and PX12 on histone acetylation and methylation levels. Coming to the effect of voinostat and PX12 treatment on histone H3 and H4 post-translation modifications. In this, we have two main categories, normoxia and hypoxia. In each category, we have four groups: normoxic control, voinostat treated, PX12 treated, and voinostat plus PX12 treated group. In an overview of experimental design, we have first performed cell culture experiments, then we have performed acid-based extraction of histones. Then we have quantified these histones and proceeded further with 20 microgram of histones with histone propionylation and trypsinization. Then we have performed desalting. Then we have performed nano LC-MS analysis. Then we have identified and quantified these histone peptides, and after statistical analysis, we have compared H3 and H4 acetylation and methylation levels among tested groups. Coming to the results, we have compared hypoxic versus normoxic groups for H3 modifications. In this, we have mainly obtained downregulation of acetylated residues when we have compared this hypoxic control versus normoxic control group, whereas we have obtained upregulation of methylated residues. In hypoxic varnostat versus normoxic varnostat, here we also obtained upregulation of acetylated residues and few methylated residues, while we have mainly obtained downregulation of methylated residues. In comparing hypoxic pH12 group versus normoxic pH12 group, here we obtained a similar pattern of upregulation of methylated residues while downregulation of acetylated residues. Here in combination group comparison, we have obtained similar pattern of upregulation and downregulation. Coming to the comparisons among hypoxic groups for H3 modifications, comparing hypoxic varnostat versus hypoxic control, here we have obtained the upregulation of acetylated residues that were previously reported to be downregulated in untreated groups. Hypoxic pH12 versus hypoxic control, we here obtained upregulation of acetylated and methylated residues. In hypoxic varnostat plus pH12 combination versus hypoxic control, here we obtained H3K9AC upregulation. H3K9AC is a loss of H3K9AC is a prognostic marker for oral cancer, and upregulation of this marker in our treated combination group is of significant importance. In comparison of H3 modification in our hypoxic testing group versus hypoxic control, here we can see that H3K9AC is upregulated in hypoxic varnostat and hypoxic varnostat plus pH12 group. Coming to H4 modifications, hypoxia versus normoxia. Here we obtained a downregulation of acetylated residues in comparing hypoxic control versus normoxic control. In hypoxic varnostat versus normoxic varnostat, here we also obtained the similar pattern of downregulation. In hypoxic pH12 versus normoxic pH12, we have obtained here similar pattern of downregulation of acetylated residues. Similarly, here in combination group comparison, here we also obtained downregulation of acetylated residues. While comparing comparison among hypoxic groups in H4 modifications, here we obtained the upregulation in our treated hypoxic varnostat group comparing to hypoxic control group. In hypoxic pH12 versus hypoxic control group, we here obtained one tetraacetylated residue that was found to be downregulated. In this combination group comparison with hypoxic control, here we obtained the upregulation of acetylated residues. Here in comparison graph, we can see that H4K16AC is found to be upregulated in hypoxic varnostat and hypoxic varnostat plus pH12 group, and it is the loss of this marker is a hallmark of cancer, and upregulation in our tested groups are of significance important. Coming to the conclusion part, PX12 synergistically enhances therapeutic efficacy of varnostat under hypoxia and Cal27 cell line, that is oral cancer cell line. This part of the study is not included in this seminar as it is focused on histone modifications. Coming to the histone modification, under hypoxia, we have found global hypermethylation on H3 and global hypoacetylation on H3 and H4, indicating transcriptional repression. H3K9AC and H4K16AC are important hallmarks of cancer, and targeting these acetylation marks by varnostat alone and in combination with PX12 under hypoxia require further investigations. This study provides clue to explore epigenetic mechanism followed either by varnostat and PX12 alone or both in combination under hypoxic tumor microenvironment. Coming to the future perspectives of this study, a combination of varnostat and PX12 should be tested in other solid cancer cell lines under hypoxic tumor microenvironment in vitro as well as in vivo. And pinpointing the mechanisms of significantly altered histone marks will provide better understanding the role of histone modifications in tested groups under hypoxia. Proteomics evolution of nuclear and cytoplasmic proteins of the treatment versus control groups under hypoxic tumor microenvironment can aid in a better understanding of underlying mechanisms. In the end, I would like to acknowledge few people, including our supervisor, Dr. Aslan Ali, a co-supervisor, Professor Dr. Syed Ghulam Musharraf, our great collaborator, Dr. Simon Sidoli, our lab colleagues, Mr. Taj Ali Khan, Mr. Syed Osama Yasin, and Ms. Tahmina Ahmed, and other lab colleagues, and my family and friends. This is our lab group photo. This is the short communication that we have published about synergism study. It includes only the synergism study part, not the histone modification part. It is about PX12 synergistically enhances the therapeutic efficacy of varnostat under hypoxic tumor microenvironment in oral squamous cell carcinoma in vitro. Thanks, and you can ask the questions.
Thanks, Rafia. So if you have any questions, please feel free to post it in Q&A box, or you can raise your hand so that I can unmute you. So I do have a question. So do you think the mechanism is because of more general histone modification change, or it's because like there's some specific subclass of genes that are being involved to explain the toxicity, and how it's the toxicity you are seeing, like, for example, the cell fitness effect in squamous cell carcinoma different in other type of cancers?
With reference to the toxicity of drugs, you are saying about the toxicity of drugs?
Yeah.
We have tested only for oral cancer, but we can test for other cancer cells also. We have done the cell viability assay for the and determine the EC50 doses for those compounds that have 50% cell viability. We have chosen that doses for our study.
But do you think the mechanism is similar? It's all because of this pan histone modification changes, or at different cancer cell types, they are going to be different from each other?
We have to study for the mechanism study. We have don't include a mechanism study in this our work.
Yeah. Okay. So I think we are going to hand the floor to the second speaker. But if you have any more questions, please feel free to type it in Q&A box, and I'm sure Rafia would be very happy to answer it.
Okay, thank you.